Let us start with the male reproductive system and its structure, and first we will study urinary bladder. So it is present here in the male reproductive system. This is urinary bladder. A tube originates from the bladder is known as urethra. This is our urethra. It carries urine. So I am writing it here that it carries urine. It enters male external genitalia called the penis. This is the penis part. Then on the side there are testes present, which are found inside a pouch-like structure known as scrotum. These are two testes. From the testis, a coil duct originates and gives rise to vas deferens. This is our vas deferens. And this coil structure is known as epididymis. The epididymis and the vas deferens are lined by the pseudo-stratified ciliated epithelium. What it is called? Pseudo-stratified ciliated epithelium. Okay. The vas deferens loops over the urinary bladder and makes a thick bulbous structure known as ampulla. So this is that thick structure, bulbous structure, and it is known as ampulla. Near ampulla, outside the urinary bladder, an accessory gland known as seminal vesicle is present. So this is our seminal vesicle. It is present on both sides of bladder, as there are two seminal vesicles we can see here. A duct arises from seminal vesicles, join the duct originating from ampulla and combines to form an ejaculatory duct. This is ejaculatory duct. And this is formed by the ducts of ampulla and seminal vesicle. The ampulla brings the sperm into the ejaculatory duct and secretions from seminal vesicles. The secretions from the ejaculatory duct is dumped into the urethra. Then there comes another gland known as prostate gland, which is single. Only single gland is present. This is our prostate gland. Now the prostate gland also secretes some item into the urethra. After this, another pea-sized gland named Cowper's gland or bulbourethral gland is present. This is also known as Cowper's gland, and it is of pea-sized. It also pours its secretions into the urethra. The bulbourethral gland is also present on both sides of the urethra. It is shown here. Ultimately, sperms and secretions from all the three accessory glands enter into the urethra and from there to the male external genitalia, where an opening is present. In biology, where there is an opening, we used a term meatus or aperture to describe it. What it is? Meatus or aperture. So basically, this opening is known as external genital meatus. What it is called, I can write here. External genital meatus, or you can say aperture, which is a common opening for both urine and seminal plasma, which is sperms plus the secretion of glands. So what is seminal plasma? It is sperms plus secretion of glands. As urethra contains both the sperms and urine, it is also known as urinogenital duct. What it is also known as? I can write here genital duct. Okay. If we see precisely, the urethra can be divided into three parts. I can write all the three parts here. The first part of urethra, where the prostate gland is present, is known as prostatic urethra. So what it is called? It is called prostatic urethra. Prostatic urethra. Then comes the part where bulbourethral gland is present, and it is known as membranous urethra. This part is membranous urethra. Membranous urethra. And the last one is the part which is lined by the penis is known as the penile urethra. If we say the longest part of the urethra is what it is? Obviously it is penile urethra, as you can see here. It is the longest part out of these three parts. Longest. Now let us talk about the secretions of these accessory glands. Let's start with the seminal vesicle first. The secretions of seminal vesicles contains very important sugars. It contains fructose, inositol, clotting proteins, and also it contributes 60 to 70% of the secretions in seminal plasma. Now the next gland comes is our prostate gland, which is a single gland, and its secretions contain calcium, phosphate, certain enzymes, and fibrinolysin. Now what is the function of fibrinolysin? It will dissolve the clots formed by the clotting proteins. The secretions of the prostate gland will be basic in nature. Why it is basic in nature? This is to neutralize the acidity of the urine. Because see, the ejaculatory duct contains urine from the urinary bladder, which make the environment of the duct acidic. Now the acidic environment is there, which is harmful to sperms. Okay. So the secretions from the prostate gland prevent this by making the environment basic. It becomes basic. Why? Because of the secretions of prostate gland. Also, the semen contains characteristic odor due to secretions of prostate gland. It has characteristic odor of semen. Sometimes this prostate gland gets inflamed, and this condition is known as prostatitis. What it is called? Prostatitis. Now come to the next, the third gland, which is the Cowper's gland, or you can say bulbourethral gland. The secretion from this gland is responsible for the lubrication of the sperms, and it is also basic in nature. Let us now study the structure of external genitalia, the penis. On the dorsolateral side of the penis, there is presence of spongy tissues. The two spongy tissues present are known as corpus cavernosa. This is corpus cavernosa. This tissue is corpus cavernosa. These are present on the lateral side of the penis. And on the dorsal side of the penis, there is tissue present that is known as the corpus spongiosum. This is corpus spongiosum. In the middle of the corpus spongiosum, there is an opening called urethral opening present for the release of the semen and the urine. This opening is urethral opening. In between these spongy tissues, there are several blood spaces or blood vessels present. Actually, during excitation, there is filling of blood in these blood vessels, due to which erection takes place and hence the insemination occurs. Now, if we talk about the tip of the corpus spongiosum or the tip of the penis, it forms a bulbous structure which is known as glans penis. So you can see here that this is glans penis. Meaning the tip of the penis is known as the glans penis, which is formed by corpus spongium only. Then this tip is lined by a retractile fold of skin which is called foreskin, or this is also called prepuce. In this prepuce, there is a gland present known as perpetual gland or the Tyson gland, which secretes a white fluid called smegma that has characteristic odor. Okay. Now the sperms in combination with the secretions of accessory glands are known as semen. The quantity of semen released per ejaculate is 2.5 to 3 ml. Per ejaculate is 2.5 to 3 ml. Number of sperms present in this amount is around 200 million. Means 200 to 300 million sperms per ejaculation are released. Therefore, in 1 ml, approximately 100 million sperms are present. In 1 ml, 100 million sperms are present. If this number is below 20 million, then the male is suffering from a condition called oligospermia. What it is called? Oligospermia. Which means low sperm count. This condition leads to the infertility in males. Now let us jump on to the next topic, that is formation of sperms. The process is known as spermatogenesis. It is called spermatogenesis. The location of spermatogenesis is inside the seminiferous tubules. Inside seminiferous tubules. It starts after puberty has reached the males. So when we studied the structure of seminiferous tubules, we saw the germinal cells called spermatogonia. What are they called? Spermatogonia. The spermatogenesis is going to start from spermatogonia. Let us study the process. Start with the mother cells that are spermatogonium. What are they called? Spermatogonium. These spermatogonia multiply by the process of mitosis, right? Then in some spermatogonia, differentiation takes place and they are transformed further into primary spermatocytes. So these are our primary spermatocytes. Primary spermatocytes. These primary spermatocytes undergo the next division, that is meiosis 1, which is a reductional division. This resulted in the formation of secondary spermatocytes, right? Now these secondary spermatocytes undergo meiosis 2 and form spermatids, right? These spermatids do not have flagellated tails and heads. These are just round cells. So these spermatids undergo structural differentiation and form sperms. This is how gametogenesis in males occurs. The ploidy level of spermatogonia is 2n as it is written here. Then primary spermatocytes are also 2n, okay, because the process by which they divided is mitosis. So the pleiade is again 2n. Next, meiosis takes place and hence reductional division takes place. Then secondary spermatocytes are of ploidy n, right? Spermatids are also n, and then sperms are also of n ploidy, right? So when the formation of spermatocyte is taking place, that process is known as spermatocytogenesis. What it is called? Spermatocytogenesis, right? After that, when the sperms are being formed here, that process is known as spermiogenesis. What it is called? Spermiogenesis, right? The release of sperms from testis is known as spermiation. What it is called? Spermiation. So these are the three important processes which you must remember. Now I'm going to ask a question. So how many sperms are formed from 25 primary spermatocytes? How many sperms are formed from 25 primary spermatocytes? Okay, so this is a very interesting question. Let's first take one primary spermatocyte, right? One primary spermatocyte we are considering, which forms, as you can clearly see, the two secondary spermatocytes, right? And out of these two secondary spermatocytes, one will form another two spermatids. That means in total they are forming 4 spermatids. So one primary spermatocyte will result into 4 sperms, right? That means if we can say that how many sperms are formed from 25 primary spermatocytes, so it is 25 into 4, which is equal to 100, right? That means 25 primary spermatocytes will form 100 sperms. So now let's study the structure of human sperm. The first part of human sperm is called as head region. The next small parts that comes just after the head is our small neck region, right? After that, middle piece comes. After the middle piece, the tail region comes, right? Now let's talk about the head region first. At the tip of the head, there is a structure which is formed by the modification of the Golgi, and this structure is known as acrosome. It consists of sperm license, and it will release some enzymes which will break the membrane of ova, and then entry of sperm contents will occur inside the ova, right? Acrosome's acrosome secretion is just like the drilling action, right? Besides acrosome, head region contains nuclear content of sperm also. So this is nucleus of the sperm. It is shown here. So how many chromosomes will be there inside the nucleus? Answer is 23. That means the condition is N condition, right? Now let's talk about the neck region. There are 2 centrioles present in the neck region, right? 2 centrioles present here. Upper centriole is called proximal centriole. Which plays important role if fertilization occurs. And the lower centriole is called distal centriole. Okay. So proximal has got major role in first cleavage. What is first cleavage? So when zygote forms, the first cleavage of zygote is because of proximal centriole only. Right? Distal centriole will help in forming axial filament. So it will form 9 plus 2 arrangement filament, which is our axial filament. It will pass through middle piece and then from the tail, right? Now come to the middle piece part. It consists spirally arranged mitochondria, and there are 25 mitochondria that are present in the spiral structure. How many? 25 mitochondria, okay? Now the function of mitochondria is to form ATP, and ATP is energy currency, you already know. So they actually generates energy and which helps in the movement of male gamete, right? Now come to the tail part. Now with the help of this region, the sperm will be going to have undulating movement. Around 200 to 300 million sperms were present in 2.5 to 3 ml of sperm, you already know. But can you think how much is actually required for the fertilization? It is at least 60% out of these 200 to 300 sperms, which should have normal shape and size, and 40% of these 60% should show vigorous motility. Only then fertilization occurs smooth. So thank you for watching this video. I hope you understand all the concepts of male reproductive system and the structures.